My name is Lauren and I'm a graduate student at the Whitney Marine Laboratory. Today I'm going to show you around the lab, tell you about some experiments that I work on, and give you some insight into what my day-to-day job is like. But before I do that, I just want to tell you a little bit about the lab that I work in. Here at Whitney Laboratory, there are many different faculty members and they each have their own lab studying a certain aspect of biology. In my lab, we study development and regeneration of a marine worm called Capitella tollida. Now, if you told me in high school that I was going to be studying worms, I probably would have looked at you like you were crazy. But the truth is, we can perform lots of experiments on worms that we can't do on other animals like mammals, and worms are really good at regenerating. You cut a worm in half, it can regrow its tail, just like a lizard can if you cut off its tail. Some worms can even regrow their heads if you cut them in half. Now, the worm that we study, Capitella, it can only regenerate its tail, but I'm interested in learning how it's able to do that and if I can figure out a way to experimentally cause the head to regrow. We're interested in studying regeneration because hopefully if we can figure out how Capitella regenerates so well, we can help humans regenerate better as well. Okay, now let's show you where we actually keep the worms that we work with. So right over here, these things that look like giant refrigerators are incubators where we keep all the worms at a constant temperature. So if we open it up, inside you can see that we have lots of bowls full of mud. That's where we keep the worms, and each bowl has about 40 worms inside. Every week, we sift the worms, which is where we count the number of healthy worms and then put them in a bowl of fresh mud, which is how they get their food. So they're really easy pets to take care of, which is important for a lab organism because you want to spend most of your time working on your experiments and not taking care of your animals.
A few times each year, the lab takes a field trip to the marsh where we collect enough mud to feed the worms for several months.
This is what an adult Capitella looks like. They're pretty small worms, but you can still see them without a microscope. An adult could probably fit on the nail of your pinky finger. The females develop brood tubes, which is where embryos and then larvae develop inside. The brood tube is like a clear layer of mucus and sand grains stick to the outside. That's what this picture is showing. This timeline up here shows what the embryo looks like as it develops inside the brood tube. Once the larvae reach this stage, they hatch from the brood tube. The larvae look like this. They have little red eyes, a green gut, and some little hairs around their neck and trunk that they use to swim in spirals through the water. The larvae eventually undergo metamorphosis and transform into juvenile worms, which are really small versions of the adult, and then they eventually grow into full-sized adults again. This is the main lab area. In it, everyone has their own lab station, and this one is my lab station. And at each station, we have a dissecting microscope to help us see the worms better. We have a bunch of chemical solutions that we may need for our experiments, some pipettes to draw up small volumes of liquid, and any other tools that we might need. One experiment I've been working on is seeing whether juvenile worms can regenerate their tails faster than adult worms, because I'm interested in seeing the effect that age has on regeneration. So to do that experiment, I first anesthetize the worms in this solution of seawater plus a chemical called magnesium chloride. And once they're relaxed, I put them on this amputation platform. Then I use this really small scalpel blade to cut the worms at the 10th segment, and I do all of that while looking at them under my dissecting microscope. Once the worms have been amputated, I put them on this glass microscope slide and then move over to a more powerful microscope, the compound scope, in order to take pictures of the area that I just cut. And I do that every day until regeneration has been completed and compare the juveniles versus the adults each day. The compound microscope, which is a more powerful microscope than the dissecting scope at our stations. I have the glass slide with the worm on it already loaded into the microscope, and then I use a program on the computer to take a picture of the worms that I can save on a flash drive and analyze later.
Once I have acquired a whole bunch of pictures, I sort them out and look at the data. This is a very important part of my job as well. When you look at your data, you may also find surprising details that you weren't necessarily looking for before. For example, I noticed that the new tissue growing during adult regeneration was a much smaller fraction of the body diameter than juveniles. We aren't sure why this is the case, so I may try to think of additional experiments to learn more about this. I also noticed that when the worms regenerate, they grow the very last segment of their body first, then fill it in with many smaller segments that eventually become full-sized. I was expecting the segments to grow back one at a time in chronological order. This just shows you how important observations are and how we should try not to make assumptions in our research. Finally, there are other responsibilities I have besides performing experiments. I am still technically a student, so I take classes too, just not as many as you all have to take. Right now I'm taking 2 classes, and so those meet just on 2 days out of the week. I also spend time going to seminars, which is when a researcher from another university or place presents about their research. I also have lab meetings, and I read research papers to stay up to date on what other researchers are doing. Sometimes there can be stereotypes surrounding graduate students where people think that we never leave the lab and we're always working hard, but the truth is that we're normal people too, and we have other hobbies and activities we enjoy, and so it's important that as grad students we also make time for those in addition to all the research that we do. I hope you all enjoyed hearing about what I do. The final message I want to leave you with is that anyone can be a scientist. The only requirement is that you're curious and ask questions. Don't ever be intimidated because you think you aren't smart enough. We keep learning our entire lives, and it's important to be patient with ourselves. In the meantime, just follow what you're passionate about and continue to ask lots of questions, and don't compare yourself too much to others. So, thanks for listening, and good luck with all that you do.